Hi, ladies and gentlemen. My name is Gary Bizignano, and I am doing neural mechanisms at play in emotional trauma. So the idea here is this lecture is going to focus primarily on the prefrontal cortex, the hippocampus, and the amygdala. These are three areas that seem to have a very, very large role in trauma, in emotional trauma, and then they have a commonality in terms of underlying mechanisms, neural mechanisms that are at play. So focusing on the prefrontal cortex, the idea here is that essentially the prefrontal cortex is in the forebrain. It is the part of your brain that allows you to perform higher cognitive tasks, of course, the complex thinking, controlling emotions, things like that. So chronic stress that results in emotional trauma is directly linked to a dysregulation of glutamate throughout the cerebral cortex. So the idea here being that glutamate could have an increased response, a decreased response in different parts of the brain. It depends on the areas that we're talking about because glutamate is scattered throughout the brain and its receptors are as well. But the idea here is we're going to be focusing for this part specifically on the prefrontal cortex. So in the prefrontal cortex or PFC, N-methyl D-aspartate receptors, NMDAR, are found in high density on prefrontal cortex dendrites. NMDA has a glutamate binding site, which allows the receptor pore to open. So basically, if you look over here at the diagram in the bottom right-hand corner, you'll essentially see a receptor. Receptors are membrane bound proteins that are designed to act as a way for things that are outside the cell to come inside the cell. And they usually have a binding site. In this case, in the prefrontal cortex, we have these NMDA receptors that are responsive to glutamate. So when glutamate binds to them, the receptor pore opens, allowing for ions to flow inside of the intracellular space of the cell. The ion in this case that I focused on was calcium. So as you can see, calcium has a 2+ charge. That means that it is an exhibitory ion. It causes an action potential, or it causes a positively charged signal, which essentially is excitation, meaning that it causes neurons to be fired. So the idea here, of course, as I wrote, glutamate is an exhibitory neurotransmitter found at high densities in the prefrontal cortex synaptic clefts, allowing for a calcium influx into the intracellular space via NMDA receptor channel. The exhibitory effect of glutamate can be attributed to calcium anions that contribute a +2 charge, which promote action potential. When the brain experiences trauma, prefrontal glutamatergic signaling is reduced, causing cognitive deficits in thinking, often attributed to stress-related disorders. Concept being that the dysregulation in the prefrontal cortex is a decrease in glutamate response, glutamate production, and essentially that part of the brain is essentially being shut down. This is one of the things that happens as a result of traumatic stress in the prefrontal cortex specifically. Amygdala response to glutamatergic upregulation. Unlike the prefrontal cortex, dysregulation in the amygdala takes the form of an excitatory glutamatergic response directly related to increase in NMDA receptor activity. So the idea is in the amygdala, something completely different is going on. The prefrontal cortex, we have a dysregulation in the form of decreased glutamate response. In the amygdala, we have an increase in glutamatergic response and glutamatergic signaling. So, well, the question is this, I guess, what form does this take? What form does this take? So the glutamine dysregulation in the amygdala, it likely takes the form of increased glutamine synthesis in glial cells from the amino acid glutamate NH3. We're going to go into that in a second. Since glutamate-activated NMDAR causes an increase of calcium anions in the intracellular space, the overexcited amygdala works with the hippocampus to encode trauma-related memories with heightened emotion. So one of the reasons why when we experience trauma, it is such a daunting memory because the emotional response from the overexcitation of glutamate causes that type of emotional response. So the psychological outcome of these electrophysiological and biochemical aspects create that emotionally perceived behavior. So in this diagram right here, I just kind of wanted you guys to understand a little bit about what's going on here. So the glial cell, which you can see is located to the left and right, that is that green blob, green diagram. You see right here that you have GLU, which is essentially glutamine going in. Then you have this amino acid called glutamine synthase, and then it produces glutamate. Glutamate leaves the glial cell and is essentially transported into the axon, where it will be packaged into a vesicle, transported to the synaptic cleft, where the axon meets the dendrite, spilled and released over the synaptic cleft, where you will see glutamate receptors are NMDA receptors are located there to allow the glutamate to bind and open the pore, allowing for influx of different types of ions. I know I said calcium, there's other things. There's also potassium and not potassium, sodium, and other ions that could influx into the channel as well. The idea right here, I put this little diagram. I just wanted you guys to kind of see, what it looks like from a chemical perspective. You have right here this carbon chain that you see right here with mostly, it's basically all hydrogen and oxygen. That is actually glutamate. And then you have right here the ammonia, NH3, essentially is going through there. It then goes into glutamine synthase, where ATP, which is essentially the energy of the body, is being used to provide energy to allow for the synthesis to occur into glutamine. Then we have glutamine right here as the final product. So hippocampal amygdala LTM. So kind of get into a little bit of feedback loops over here. The idea is the hippocampus is the structure of the brain that is responsible for encoding long-term memories. The effects of glutamine on the hippocampus are mixed and highly dependent on the region. Generally, the hippocampus creates memories that have an emotional response created from a glutamatergic-dependent excitotoxic amygdala. So the idea here being that overstimulation of the amygdala actually takes a form of excitotoxicity, meaning that too much glutamate output can actually damage the brain. And this is why, trauma-related emotional trauma actually can have a negative effect, where it can deregulate cerebral density and decrease the amount of neurons in the brain due to the fact of, just simply put, neural cellular death. Keep it simple. These memory traces are potent and can have a repeating effect on memory recall. So again, because these memories are being stored with such an excitotoxic response from the amygdala, it makes the memory so strong, and it makes it repeat, and it can repeat over and over again as a result. Now, it doesn't mean that the brain can't heal, but the idea is, for some trauma, this can have a lasting effect. So trauma evolutionary benefits. So the idea is that, yes, trauma occurs because there likely has been an evolutionary benefit in the past. To understand the potential benefits to trauma, we definitely need to take an evolutionary approach, as we do for most things in terms of behavioral deficits that we might see. Trauma likely developed as an emotional survival response in our evolutionary descendants. Hypersympathetic and parasympathetic nervous system responses to events can create the psychological effect of heightened negative emotion that is the result of biochemical electrophysiological responses to severe stress that can enhance survival odds when encountering similar situations. Again, simple sympathetic and parasympathetic responses can create a stress-like environment when we're dealing with emotional trauma. Those things are heightened. In the case of sympathetic response, you have a hypersympathetic response and a hypo, meaning a shutdown of parasympathetic responses. And what happens in this type of situation is it allows you to have, from an evolutionary perspective, if you had a very traumatic event happen in the past, it can increase your survival odds, because now you know without a doubt that event happened, and there's such an emotional attachment to it, to make sure that doesn't happen again. I chose right here a picture of archaic humans, Homo heidelbergensis. They're a species that predates the modern day Homo sapiens sapiens, and they are hunting a Megaloceros. Megaloceros is basically a megafauna, a very, very large deer that is extinct today. But I thought that kind of did a good job of seeing how if one of your if one of the group mates, essentially one of the tribal members, died while hunting one of these animals, then that could create a traumatic event where they may decide not to hunt an animal like this because of the stressful event that had been created as a result, resulting in, yes, a type of trauma, but one that could work for the benefit of survival of the species. So in summary, here's what we basically have here. The prefrontal cortex exhibits a decrease in excitatory activity as a result of decreased glutamate output and at the synaptic cleft, creating a depressive mental state. The amygdala sees an increase in glutamine-dependent NMDA receptor activity that causes overexcitation of the amygdala, creating the psychological effect of intense emotion. The emotion is then encoded into long-term memories about the hippocampus. The excess glutamate output creates an effect known as excitotoxicity, where overstimulation of certain neural circuits results in brain damage. Studies have shown people with severe emotional trauma, they tend to not make the best decisions. And this can likely be attributed to the excitotoxic response of glutamate.